Hello everyone. Today we are going to start a new topic, that is asexual reproduction. Already you all know what is asexual reproduction. It is a type of reproduction in which only one parent is involved, right? No gamete formation takes place. Gamete means no sex cells formation takes place. There are no sex cells required in this. One parent only gives rise to the new daughter cell or new offspring. Now this asexual reproduction, it is of 5 types, maybe not 5 but 6 types. Vegetative propagation also comes in this. First is fission, then comes fragmentation, regeneration, budding, spore formation and vegetative propagation. So we are going to study all these 6 types of asexual reproduction one by one, right? So the first one is fission. Fission actually means breaking up or splitting, right? So this fission, it is of 2 types. Binary fission and multiple fission. Binary as the name indicates, bi means 2. Splitting up into 2 daughter cells. One organism splits up into 2 daughter cells. And multiple, of course, means number of cells are formed in this. Binary fission, the most common example is amoeba. Most of the bacteria and protozoans, that is the unicellular organisms, they reproduce by this method, that is binary fission. So in binary fission, if we take the example of amoeba, you know, when amoeba, it does not have any fixed shape. When it is completely mature, ready to divide, it takes almost a round shape. The pseudopodia, they are not much protruded out. Otherwise, you know, the amoeba, mostly these pseudopodia, they are protruding outside. But when it is mature, when it is ready to divide, then it takes almost a round shape, right? Now what happens, a constriction starts appearing in the center and the nucleus also starts elongating. Then this constriction, it goes on deepening and the nucleus, it ultimately divides into 2 parts and the cytoplasm also starts dividing. They start gathering around the nucleus. The cytoplasm starts becoming more dense. Then ultimately this constriction goes on deepening and the amoeba, the parent amoeba, it divides into 2 daughter cells. From one amoeba, 2 new cells are formed. 2 new amoeba are formed. This is known as binary fission. This takes place in most of the protozoans like paramecium. Paramecium also the same thing happens. First the nucleus divides into 2 parts and then the paramecium divides. You know, paramecium, it is a slipper shaped organism, right? In the shape of a slipper. The slipper that you wear in your foot, right? And it has a nucleus here. So the nucleus, it divides into 2 parts. One nucleus goes in the upper part, the other goes in the lower part, and then it divides into 2. Then there's another example of a protozoan which is called Leishmania. Now this Leishmania, it has got a whip-like structure here, right? And a nucleus here. Now this Leishmania, it always divides like this in a fixed plane, in a fixed orientation. Amoeba, it can divide, like the constriction can appear this way as you have seen in the example, and it will divide into two amoeba. Or it can also happen that the constriction starts appearing in the center like this, and it divides into these two parts, one above, one down. So in amoeba, the fission can take place at any plane, because amoeba does not have any fixed shape. So it can take place horizontally, it can take place vertically, it can take place diagonally, in any plane. But in Leishmania, it always takes place in a fixed orientation. This Leishmania, 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 it causes a disease known as Kala Azar, or it is also known as black fever. Right? So this question comes in the exam sometimes. What is the main difference in the binary fission of amoeba and Leishmania? Right? So you have understood, in amoeba it can take place in any plane, while in Leishmania it is in always fixed orientation. Now next we take the multiple fission. Multiple fission, the best example is Plasmodium. It is a malarial parasite. Right? Causes malaria. Now in this, what happens in Plasmodium? This cell, this is a cell present here with nucleus and cytoplasm. During unfavorable condition, when the conditions are not favorable, the food is not available or the correct temperature is not available, it develops a protective wall around it. And this protective wall, this is known as cyst. And inside this nucleus, it starts dividing like this into many cells. Right? From one cell, many cells are formed. Each cell will have its own nucleus and cytoplasm. And when the favorable conditions will return, this cyst wall or the protective wall, it will break open and all the multiple number of cells are released out. So from one cell, multiple number of cells are formed. This is known as multiple fission. Next type of asexual reproduction is fragmentation. Now in fragmentation, we'll take the example of Spirogyra. The main difference between fission and fragmentation is that fission, it takes place in unicellular organism, while fragmentation, it takes place in multicellular organisms. But these multicellular organisms, they also have a simple body design. Now what happens in case of Spirogyra? Spirogyra, it has a spiral structure. And here in each part, you can see there's a nucleus plus there's cytoplasm and the other cell organelles also. So what happens when this is completely mature? At that time, as the name indicates fragmentation, it breaks down into fragments. Right? It breaks down into fragments. And each fragment, because it has nucleus, it has cytoplasm, it will develop into a mature organism. Right? So from one parent cell, one parent organism, you can say, from one parent, many new offsprings will be formed. Right? As many number of fragments will be there, the new organisms will be formed from that. Right? But mainly, you can say that why it does not happen in all the multicellular organisms. Even we are multicellular organisms, but as I told you, fragmentation occurs in the multicellular organisms which have a simple body design. Like in the complex multicellular organisms, the different cells, they have different functions to perform. These cells, they will combine together and form the tissues. Tissues will form the organs. These organs will be placed in different parts of the body, performing different functions. Organs will form the organ system, and there are some specialized cells which are meant for reproduction. All the cells cannot take part in reproduction in the complex multicellular organisms. So this can take place only in the organisms which have simple body design. Next comes regeneration, to regenerate the body parts, to form new body parts. Now in this, what happens is, here we are taking the example of planaria, right? Planaria. Planaria is also known as flatworm. Now this is also a multicellular organism, but this also does not have a complex body design. Now here what happens, if somehow any body part is cut, like here I have shown this, suppose this head is cut, or the middle part is cut, or the tail part is cut. What will happen? This head, right? Suppose this is the head. It will develop the middle portion as well as the tail portions. What happens is, the cells here, from where it was cut, they start growing and developing, right? They form a group of cells, and these cells, they form the different body parts, and they start functioning there and form the whole body organisms. Or the middle portion, it will form the head part also as well as the tail part also. Same way, the tail will develop the other two parts, right? Now what is the main difference between fragmentation and regeneration? In fragmentation, when the organism is mature, it by itself break up into fragments and develop into new organisms, right? In regeneration, it does not cut its body parts on its own. It is accidentally, if any part of the body is cut, then it will regenerate its other body parts and grow into a new individual. This will grow into a separate new individual. This will grow into a separate new individual. So from one organism, if any part is cut, that will grow its new individual organism. And this will also develop. Suppose only the head part was cut, this whole organism will develop the head part and start growing. And this head part which was cut, that will grow the lower portion and start developing as a new organism. So this is known as regeneration. Regeneration also takes place in Hydra. In Hydra also, if any body part is cut, then that part can be developed, right? Hydra also has got this ability to develop the cut part. Now next is budding. Now budding, as the name indicates, bud, formation of a bud, like in case of Hydra. Now in case of Hydra, what happens? You know, Hydra, it has got tentacles on its mouth part. From any part of this stem or its body wall, what will happen? A new bud will grow. This bud will keep on maturing, develop the tentacles also, right? And when it is completely mature, it will detach itself from the parent part, and this bud, now it will grow as a new Hydra, right? and then from this hydra also the new bud will grow, that will also mature and develop again into a new hydra. This budding can also take place in yeast cells. In this, what happens? Multiple number of cells are formed within no time. Now this yeast cell, it is a kind of, you can say, oval type of cell, and it has a nucleus and cytoplasm. So what happens? The cytoplasm, it starts growing at any one place, it becomes very, very dense, right? So this dense cytoplasm, it then starts developing a bud-like structure. And this nucleus, this also breaks into two, and one of the part, it will slip into the bud. And this will then detach itself and develop as a new individual yeast cell. This is one way. Otherwise, sometimes what happens, this bud, it will not detach itself, but it will develop another bud. This will then mature and develop another bud on it. From one yeast cell, from many parts, the new buds will start growing, and they will keep on accumulating, and then a stage will come then when all these buds, they will detach themselves from the parent plant and grow as new individuals. So this is how this bud formation will take place, right? Now the last one is, not the last one, but the fifth one is spore formation. Now what happens in spore formation? Like in the case of, we can take the example of bread mold. Now, in case of bread mold, if you take a bread piece and just wet it a little, just sprinkle some water on it and keep it in some dark place. After 2-3 days, you will see that a white color fungus is grown on it. And then if you keep it for some more time, you will see some black dots on it. Now if you just take one thread and see it under the microscope, you will see the structure of this type. Right? These are root-like structures. These are known as hyphae. And this is the stem, and then a round blob-like structure on it, and in these the spores are present. This round part, this is known as sporangium, in which the spores are present. These spores, you can say, they are the seeds from which the new hyphae will grow. So what happens during unfavorable condition, this sporangium also develops a cyst wall or a protective wall around it. Now what are these unfavorable conditions I'm talking about? Unfavorable conditions means when proper nutrient is not present, right? They do not get proper nutrients. Then you can say moisture is not there, or appropriate temperature is not present. Then they develop a cyst wall, a protective wall, and these sporangium, they go on multiplying inside this protective cyst. And when the favorable conditions return, when they have the moisture available, the appropriate temperature available, the nutrients are available to it, then the cyst wall, it breaks open and all the spores are released out. Then these spores, they are in multiple number, and they go and find some other suitable substratum, and on that they get fixed and start growing again. So from one sporangium, many spores are released, and many new plants can be grown, right? So this is how reproduction takes place through spore formation. The last one is vegetative propagation. Now, vegetative propagation means when reproduction takes place by the vegetative parts of the plant. Vegetative parts of the plant means like roots, stems, leaves, etc. Seeds, etc. are not used for further reproduction, right? So vegetative parts means roots, stems, leaves, etc. Like if we take the example of potato, right? On the potato you must have seen there are certain black color spots on it. Now if we take just one portion of this having this black spot, this is known as eye. This is called as eye. And we put it in the soil, in the moist soil, and after a few days we will see the roots as well as the shoots coming out of it. Right? This is how the new potato plants are grown. Or through leaf, if we talk about, there's a leaf called bryophyllum leaf, right? Now, in this bryophyllum leaf, in the notches of this, some buds start developing, right? Small buds starts developing. And suppose these, now when these buds, they are mature, suppose they fall down in the soil which is moist, they will there develop roots and they will start developing into a new plant. So from the leaf, new plant will be grown. Now this vegetative propagation, it also takes place by artificial means. Now what is these or this leaf example we can take also of the money plant. In money plant also, if you see, suppose this is the plant growing and these are the leaves growing here, right? If we take any part, here suppose we cut this part. Suppose we cut this part, right? And we grow. So what will happen after a few, now if you put it on here in the soil, right? We'll see that the roots will start developing and new leaves will be formed here. But there should be at least one bud here. If we take this portion, suppose we take this portion where there is no leaves growing, this is known as internode, where the leaves grow or the new stems come out, that is known as the node. If we take this portion and put it under the soil, no new plant will be formed because there is no node there. The growth will take place only if there is at least one bud in the stem, right? So, money plant also can be grown by this method. Then we are talking about the artificial propagation. Artificial propagation, we'll discuss three types in this. Layering, grafting, and cutting. Now, what are these three? Layering. Layering, like we can take the example as in the case of jasmine, lemon, strawberry, raspberry, all these plants. Now what is done in this? Suppose there's a plant growing here with the stems coming out like this, right? A plant growing like this. Now this stem which is near to the soil, that is taken and bent like this and here put under the soil. From this side also this can be bent and then it is put under the wet or moist soil. After a few days, what will happen? The roots will start developing here and a new stem will come out, right, from here also, and a new stem will come out and start growing, right. Then this branch which was from the parent plant, that is removed, that is cut or detached, right, and a new plant will be formed on both the sides. Then we can again take the stem of this, bend it and put it under the soil. So like this way, from one plant, many plants can be grown. This is known as layering, right. Next is grafting. Now in grafting, what is done? A plant is taken which has roots deep into the soil, right, and here what happens is actually, suppose a plant is not giving us the desirable quality what is required. Maybe the fruits are not very juicy, or the quantity or the quality is not very good. So what, but the plant is very strong, right, the stem is very strong. Now the stem of this part is taken. This is known as stalk, the stem which has got roots under the soil, and a stem of another plant which gives us all the desirable characteristics which are required, juicy fruit also, and good quality also, good quantity also. The stem of that plant is taken, right, and that is also cut in a slant way, and both are joined together. They are combined together tightly, and on that a polythene is wrapped and it remains there for a few days. What will happen? The cambium of both will combine together. The function of xylem and phloem will start taking place, and what will happen now? We get a new plant, right? This is known as scion. This stem which is attached to it of the new plant, that is known as scion. Now the stalk and the scion combine together to produce all the desired qualities. Whatever was required, maybe juicy fruit and good in quantity, good in quality, etc. So a new plant can be formed. This is known as grafting. And cutting, as I already told you, as is done in the rose plant, money plant example we took just now. In rose plant also what is done? The stem is taken, and that stem should have at least one bud, right? And then that stem, then it is planted in the soil and we get a new rose plant. But there should be at least one bud in it, right? So these plants we are not growing through seeds, right? So there are many advantages of vegetative propagation. Advantages: one is that we can grow the plants in multiple number, right? From one plant, many plants can be grown, as we saw in the case of layering also, right? Cutting, money plant and all. From one plant, many plants can be grown. So a number of plants can be grown. Then the plants, the new plants which we get, they are exactly, genetically they are exactly same as the parent plant. Then the plants which cannot be grown by seeds, orange, banana, etc., they can be grown by this method, right? So there are so many advantages of vegetative propagation, right? These plants, they do not need much attention also. They are in the early years, they do not need much attention. So these are the advantages of vegetative propagation. Then there is one more method by which the plants are grown, that is known as tissue culture. Many ornamental plants, they are grown by this method. Now these days in the marriage parties or the parties, you see lots of flower decoration. So these flowers, they are mainly grown by this process. The orchids, the chrysanthemums, the other ornamental flowers, they are mainly grown by this method. Now what is done in this method, the meristematic tissue of the plant is taken, whatever plant we need to grow. The meristematic tissues in class 9 you have read, they are present at either the tips of the roots or tips of the shoots. The growth takes place in meristematic tissues only. So the meristematic tissue, it is cut from any plant and then that is put in some artificial medium. Now this artificial medium, it has all the nutrients which are required for the growth of the plant or the growth of this tissue, right? All the nutrients are present in this artificial medium. Appropriate temperature is also provided. Whatever nutrients are required, that is provided to these tissues, and these tissues then they start dividing into different cells, and these cells, they grow into an unorganized mass which is known as callus. Callus is actually an unorganized mass of cells. Right? What does this mean? Unorganized mass of cells means there is no differentiation here. It is not that this has grown into roots, shoots, leaves, etc. and the plant has started growing. Just an unorganized mass of cells. A group of cells is formed. Then to this callus, then again plant hormones. Right? Plant hormones are provided which are helpful in its growth and differentiation. Now, what is the meaning of differentiation? Differentiation means differentiating the plants into different organs like roots, stems and leaves. They are differentiated, right? So, what will happen when these plant hormones will be provided to callus? Small plantlets like these roots will start growing and small shoots will start coming out, right? Small plantlets will be formed like this. So these plantlets, now they are taken and either they are grown in the, they are not grown, but you will say they are planted in the flower pots, or they are taken to the nurseries and planted in the soil, right? And planted in the soil. So from just a small piece of the meristematic tissue, thousands of plants can be grown from these cells, from this callus. Many small plantlets will come out, and these plantlets, then they are grown in the soil, in the nurseries, and from that many plants can be grown, right? So this is known as tissue culture. So this was all about asexual reproduction, and now next time we'll take up the sexual mode of reproduction. Till then, keep safe. Thank you.